This is the Shahid Shah Show. Straight talk on building, buying, and investing in digital health. No hype, no buzzwords, no speculation. Just what actually works, maybe what doesn't, and most importantly, why. If you're building healthcare technology or buying it, this is where reality shows up. Welcome to the Shahid Shah Show. Today I want to tell you about a report. Not a patient, but a report. A patient gets a biopsy. The tissue goes to a lab. Weeks later, a document comes back to the oncologist, something we've seen probably many, many times. Pages of genes and variants and percentages, footnotes. Somewhere in that document is the one line that might change a treatment. And somewhere around page 11, most of us would start skimming, but of course, physicians have to pay attention to all those pages and all the documents that they're coming out with. And this is where we wanted to start the conversation, because I think a diagnostic like that is not really just a science product. It's a decision product. That sequencing, the assay, the lab work, all of that exists for one reason, and that's to help a human physician or group of physicians make a decision faster. Now, I talked about this in a couple of episodes ago, where I named that problem decision debt, when organizations take time to make a decision. So decision debt in a hospital IT governance committee, like I discussed in my previous episodes, is kind of bad. I mean, it costs money, it costs morale, and maybe costs a few quarters of momentum. But really what I want to focus on today is decision debt in an oncology treatment plan. That's a whole different animal entirely, because the person carrying that debt doesn't have a few quarters. Cancer does not wait for your tumor board to reconvene. Time is the one input nobody gets to renegotiate. And every day we spend waiting on an unclear or incomplete answer is a day that debt compounds against an actual person, not a budget line. So today's question is really decision debt wearing a lab coat. Does reading a tumor's entire exome and transcriptome instead of a curated slice of it actually pay down that debt faster? So we've seen this, and for decades, that report that we see for oncologists was built from a panel, usually a curated list of a few dozen or 100 genes that somebody was deciding were the important ones. It was a reasonable choice in the past, except today, we've got much more capabilities. So an entire company was built on a different bet. Instead of a curated list, read essentially everything, whole exome, whole transcriptome, DNA, RNA, both, and let the clinical relevance catch up afterward instead of trying to predict everything in advance. That company that we're going to cover today, and we're going to do it in a couple of episodes coming up as well, is a really exciting one called Caris Life Sciences. And as of the most recent count, they've run that bet across more than 1 million molecular profiles. Now, you can learn a lot over 1 million of anything, but we all should be suspicious of big numbers like that, because a bigger haystack, we've learned in medicine, is not necessarily going to help us make better or faster decisions. And that's why the person we've got today, really excited to have Dr. James Hamrick, chairman of the Caris Precision Oncology Alliance, which is a network of cancer centers. They actually use this data in real patients to come and tell us how valuable that data is at that quantity, at that scale. Before running the Caris Oncology Alliance, he ran clinical strategy at Flatiron. Before that, he was practicing oncologist at Kaiser Permanente, Georgia. He's been a builder, a buyer, a clinician, in that order. And of course, that's what we cover. What do builders build? What do buyers buy? And that resume is exactly what we like to have on the show. So James, welcome to the show.
Shahid, thank you so much. It's a delight to be here, and I really love the framing you set up there around decision debt. I'm very passionate about enabling really busy oncologists to make a better decision, and that's a theme that's carried through all the different scenarios that I've worked in. So great to be here.
Awesome. And what I loved about your past is you've been that product person at Flatiron, and you've been the oncologist actually seeing patients at Kaiser. Maybe let's talk about a moment where you think you got the clinical need wrong. Everybody makes mistakes, so wrong here doesn't mean you necessarily are looking back on something over and over again. But did it give us one example on where some data was not existent and you made somehow a clinically invalid decision that got corrected with later data, something that changed your mind?
I think the way I'd like to, so there are any number of areas where I've learned from a prior mistake and happy to chat about those. But one of the foundational ones back when I was leading a team of oncologists in clinical practice, and we were thinking a lot about quality and how can we ensure that, every cancer patient that comes in, if they see one of us, they're going to get a high quality experience, no matter who they see. We used to spend a lot of time curating these sort of reports for clinicians. They were designed to be fed on a regular cadence to the physician, and they would talk about everything from, did you stage the patient and document that? Did you complete all of your orders on time? Did you provide all the documentation that it's going to need for the patient to get care from other consultants? We would curate these reports and we would send them out over email to the physicians. And we were shocked. I mean, shocked when we discovered that they weren't reading them and they certainly weren't actioning on them. And our PDSA cycles weren't effective. And I learned what I learned there. Um, I relearned again when I moved on to Flatiron. I was working within the EHR, which is busy clinicians. You have to meet them where they are in their workflow. Workflow matters. If you want to impact their behavior, don't throw a lot of data, no matter how good it is. A really smart product manager told me this once. He said, "You don't build a new highway, you pave the cow paths." And what that meant is, you build things that fit into the clinician's workflow where they're already going. And so I learned that if you want to feed data back, quality data back to doctors, decision support data back to doctors, you got to try to put it into their EHR workflow or some other way where it really meets them where they are. You can't expect busy doctors to go out of their workflow because your little project is so important. And that is a theme that has come up again and again. Things like surfacing clinical decision support for clinicians. It has to be correct. It has to be served up at exactly the right time when they're making the key decision. And it's a huge opportunity when you do that. But no matter how good your tool is, if you don't meet them where they are, you're going to fail most of the time.
Love it. Yeah. In fact, paving the cow paths really, if you look at the long term, paving the cow path gets you started, but really you guys have gotten much further than that. Now, why do you think in the past the industry defaulted to targeting gene panels for so long when
Yeah.
everybody building them knew that it would be stale on arrival?
Yeah, so I'm really lucky. I'm getting old now, but I'm old enough to remember when I was a resident at UC San Francisco in my continuity clinic, which is where they give the residents a panel of patients. I had a patient who had been part of the clinical trials for a drug called Gleevec or imatinib. This is one of our first targeted therapies for a disease called chronic myeloid leukemia, based on the BCR-ABL, which is a biomarker, a genetic alteration. And this is a patient that I will never forget because they probably would not have survived to be in my clinic had this targeted therapy, biomarker-driven therapy not existed. So I was able to watch, we moved from that where we had single biomarkers to begin to differentiate different types of leukemia, different types of lung cancer. It used to be lung cancer is lung cancer. Then we had small cell, non-small cell. Suddenly, the early 2000s, we start getting into EGFR positive, ALK positive, ROS positive. These are all different genomic alterations that you can find. And we built to the state of the science. So, measuring these individual biomarkers were huge breakthroughs that were the state of the science, and they were what we could do through immunohistochemistry, through polymerase chain reaction, those types of technologies. And so the science followed along. So it wasn't really a question of us having the luxury of doing relatively low cost broad sequencing. It was here are the tools we have at our disposal. I also am old enough to remember the Human Genome Project, where it took billions of dollars and 13 years or so to sequence the first genome. I can now stand, in front of a machine that can do 196 full genomes in 44 hours, at a remarkably low cost. So it's really been a case of the technology and the cost meeting the moment scientifically. We've proceeded from single biomarker, so that might be EGFR testing in non-small cell lung cancer, to small reflex testing where, okay, if this test is negative, I move to that one. And then moving forward to panels. Now, remember, and lung cancer is a good example. We got out to 7 or 8 different biomarkers that you needed. And at that point, you're not running out of biomarkers, you're running out of tissue. So remember, you have to run these. If you're reflexing and doing individual a la carte testing, you're burning a little bit of that extremely valuable biopsy specimen every time you do it. So, at some point it becomes that if you can run next generation sequencing and do sort of parallel broad high throughput sequencing, you're just going to get a lot more data with the tissue that you have. That matters to patients because it's more answers they can get. These biopsies are often invasive. Going down with a bronchoscope, that's not a small thing when you're a patient. Putting a big needle into your liver, that's not a small thing. So, I think it was basically the technology and the science, and what was possible in terms of the broad sequencing. Um, and now we've got NGS panels where initially we would do maybe a limited scope. We can talk about it in a minute, but there sort of becomes a philosophical, are you chasing what the tumor knows about its own biology, or are you chasing the biology itself? What do we know about the tumor is sort of the limited version where you're just looking for the biomarkers that we know versus what does the tumor know, which is the universe of biology that's driving these cancers. So
No,
broader becomes better.
no, what I loved about that is that I think you hit the nail on the head with the idea that you got to use the same or less human parts, tissue,
Yeah,
and other biopsy data. Not just the output has to be much, much bigger, but that output has to be bigger from a smaller piece of input. Fantastic. So in that
yeah.
case, what does it actually take to operationally deliver the whole exome and the whole transcriptome sequencing at a national scale? Sounds simple, but you got to ship this at high volume. What does that mean? And
Yeah,
why are
yeah.
others not
So
able to do this?
yeah, it's a great question. It's a big undertaking. And so first of all, the whole exome, that's all of the coding DNA. So that's a big bite. And then the whole transcriptome, that's all of the RNA. And each of these give you a sense of what is the biology underlying this tumor. So to do that on everyone, the decision was made sort of philosophically at Caris that that's the best way to follow the biology. You really have to have an operating system. And it took me time to understand this, even as a medical oncologist, until I got to Caris, I didn't really fully understand it. So you need logistics, you need a wet lab, you need a bunch of pathologists with pathology expertise. You need to be able to do tissue handling. You need to be able to do the sequencing. So you need the equipment that can run this sequencing. As I said, remember the Human Genome Project was recent enough for me to remember it. And so you need the hardware to do the sequencing. But it took me, and I think it still takes a lot of clinicians a while to realize just having the sequence, a string of A, C's, T's, and G's on a huge piece of memory somewhere, that doesn't do much for you. So the entire specialty of bioinformatics is using computing and analytic power and human expertise to make sense of all of that. The genetic code is incredibly long and complex, and so you need an operating system with a lot of experts that allow you to create and understand your bioinformatic pathways, so you can provide some actionable insights to the people that care. So, as has been a theme here, it's not about delivering more data; it's about delivering things that actually impact decisions. And so, to do this at scale really requires that operating system, I like to call it, if you're going to make it clinically useful. And doctors don't need another 1,000 variants. They need the ones that matter. What can I do for this patient in clinic when I see them again? And they need
Right.
them quick. They need them quick. That's the other piece is there is a patient on the end of that. I've sat in the room with the patient. I've been that doctor that's having to say, well, we would like to know how you're going to do. We would like to know your first line treatment, but we're waiting on a result to come back to tell us about your tumor. So you got to do all that quickly.
Yeah. So if you're just tuning in, this is the Shahid Shah Show, where we usually cut through the noise in digital health, but today we're extending digital to also mean bioinformatics, molecular, and other types of health, and to talk honestly about how healthcare technology actually gets built, deployed, and paid for. Today we're in the first of a three-part series with Caris Life Sciences, where we're asking whether comprehensive molecular profiling actually gets the right treatment decision made faster, or is it just another longer report and the clock keeps running? What I'd love for you to do, James, is think back to your days as an oncologist. what did you do differently, and what would an oncologist using the new Caris data do differently with the newest reports in terms of patient care, maybe in a tumor board? How does this stuff change that workflow?
So you have a lot more information at your fingertips, which can be a blessing or a curse. And so the ideally your report is coming to you with some good insights baked right into it. And one of the first things I can say is I think you can be a little bit more comfortable. So one of the functions we would all want to get out of artificial intelligence is to have it sort of as a safety net, not replacing our doctor or our pathologist, but kind of double checking that there's not something being missed. So we now there are a number of patients that come into the cancer clinic that you do a biopsy, it shows cancer, but you don't know which organ it started in. So maybe it's a liver mass, and you don't know, did it start as a colon cancer or a lung cancer? It's a real problem. It's called carcinoma of unknown primary. There are types of tests now that can be run in parallel because we have all the whole exome and all the transcriptome data. We can use bioinformatics and artificial intelligence to compare that tumor to a large data set of colon cancers, a large data set of lung cancers, a large data set of breast cancers. And what that can do is suggest a tissue of origin. And so, this is something that you can do because you're doing whole exome, whole transcriptome. You can build a model and an algorithm to help to suggest, hey, this disease, that if it's a breast cancer, you're going to treat it this way. If it's a lung cancer, you're going to treat it that way. The outcome matters. We're now doing some really high level analytics based on the genomics to help you make that decision. In addition, so that happens maybe 1%, 2% of the time in all solid tumor patients. In addition, in the background of all cases, this runs. GPSAI is what we call it. It runs, and we will actually, if it is discordant from the diagnosis that the physician has submitted, so if they said, "Hey, this is Mrs. Jones, she has a breast cancer," and it says, "Hey, wait a second, we actually think this is a blood malignancy," we then begin a conversation between the pathologist, the ordering physician, to arrive at the right diagnosis. And so knowing that that's working for me in the background is something that is very different from what was happening earlier in my career.
No, you pointed out something really unobvious here, which is typically the oncologist is supposed to know what they're looking for. They go
Yeah.
biopsy the thing that they're looking at, then they get a report on it. But what's really cool about what you're talking about is it can give you a GPS-like feel, like
Yeah.
it gives you the map exactly, and then you can map where you're going. I actually didn't realize that until you just mentioned that here. Now, given that this is a lot of data coming out, what is the maybe the training is the wrong word, but what is the education, the training that you have to give to the physician so they use it the right way and maybe don't apply it to things that can't be reviewed using the kind of report you're giving? So what are you seeing in the marketplace as they're using it?
Yeah, it's a great question. So I will tell you, medical oncologists, we don't get as part of our training a PhD in genomics. And that's almost what you need in this day and age because the biology is so complex and all of these principles of molecular sequencing and genomics, most medical oncologists, unless that's your area of research, are not fluent in that. And so I like to say sometimes when I'm speaking to a group of medical oncologists, I'll say, what does it mean? What does the whole exome or the whole transcriptome even mean to you? And you can tell there's a pause and this is exactly how I was until I really dove deep here, where it's like I learned that, I know what it is, but it's almost like listening to a different language. You're not fluent. You have to take a pause for a second. And so what that means is this stuff is very complex. It's very nuanced. It's our job, and we put a lot of thought into this to make our results and our reporting as interpretable and actionable as we can. And so we put a lot of thought into we have a team of PhD genetic experts and clinicians, et cetera, who put a lot of thought into the way things show up on our report so that you get the facts, you get everything you need and not one bit more. So that becomes an actionable insight in the clinic. And so I think one of the important things is to realize, you really need to think about with everything we do, whether it's the report that we produce at Caris to enable the best therapy at the right time, or some other tool to put treatment guidelines in front of someone. really serving it up and setting the doctor up for success, where they don't have to, it meets them in their workflow where they are. They get the actionable insight. You're not overrunning them with data, and they can make a really good decision for the patient and move on.
Yeah, so a couple of things is that if you think about it from the chief financial officer's position, the chief executive officer's position, or the chief information or chief medical informatics groups, that C-suite has been empowering physicians with technology for many years. Often it is a device,
Yeah.
often it is a new EHR. Where do you think this precision oncology should fit in terms of the selection of technologies in priority order? And then how would you suggest the C-suite look at this and say, hey, if you're not doing precision oncology, here's where you're leaving a bunch of money on the table or a bunch of potential errors on the table. Talk about it from the business end just a little bit
Yeah.
to let buyers understand what to buy.
I think you come with the core belief that you have to understand as much as you reasonably can about the bio when you're treating a cancer patient, about the biology of the tumor to give the patient the best treatment. We're all in this game because we want to do the right thing for patients. We all ourselves or our loved ones have had cancer. We know all of that. We live in an era where you want to get to the best treatment. You want to empower your doctors where they are in their workflow, so usually in the EHR or easily accessible with all of the information they need. And in the world I live in precision oncology, that means that they've got a test that has overturned every reasonable stone to understand what's driving this tumor because picking the right first line therapy can result in much less utilization of the ER and putting patients in the hospital and subsequent lines of therapy, et cetera, et cetera. And I think that's a principle that's been hard won throughout the field. Some of this stuff is very hard to study, but there are a lot of good studies out there around utilization and selecting the right therapy. There are a lot of missed opportunities. So, we published some data when I was at Flatiron that showed that there are a lot of cancer patients in the United States that are not getting optimal therapy based on their biomarkers. And it's a systemic problem more than anything. It's not a bad physician, lazy physician, unmotivated patient. It's stuff is moving very fast. And you need to embrace the things that are going to deliver the best understanding of what's driving this tumor so you can pick the best therapy. There's a new FDA approval for a cancer drug literally just about every week. A general oncologist like myself, you can't read all the journal articles. You're too busy doing your real life, your real job. And so it's really important to set your docs up, and that will, if you focus on having the best patient outcome you can have, that's going to drive everything else in your business. The a hallmark of excellence in oncology care is things like are they giving the most up-to-date therapies? Are they using the tests that are going to reveal the biology? Do they have cutting-edge clinical trials available? You do those things and you stay at the bleeding edge, and follow the science. Word will travel. You'll have more business, and you'll use your scarce resources better. You don't want to clog your ER up with preventable side effects. It's terrible for your brand, and it's most importantly, it's bad for your patients, bad for your doctors too. So
No, I mean, if you think about what you just said, what is terrible for your brand is if you don't know where the source of the cancer is. So you're doing a
right.
bunch of biopsies, et cetera.
Oh,
But if you can do one data call, for example, from Caris and say, I can tell you where the cancer started, and then say, not only that, but that same tissue pull that you pull, but that you biopsied can then tell you what the most likely,
right.
most effective first line therapy is. Those
Yeah.
are both brandable at the hospital. Like, come to us because we'll tell you where your cancer started. Come to us because we're going to give you the best first line therapies. I mean, those should be pretty easy for anybody to think about. But it
Right.
seems like, James, that maybe physicians don't always understand precision oncology. So where do you think they might be misunderstanding precision oncology about maybe the past is, that it wasn't good enough in the past, but now it is?
Yeah.
Or maybe they're
Yeah.
applying in the wrong place.
I think understanding the power of comprehensiveness is a real opportunity. So looking, you might not realize that a limited panel that looks at 300 or 500 specific genes is different than something that looks at the whole exome, which is, 22,000 genes. You might not understand that a limited RNA panel, you may not understand that RNA adds a whole nother lens to opportunities to find treatments that may help your patient get a better outcome. And then within RNA, looking broadly may reveal signals in the tumor's biology that may get them a better standard of care option, or may, if you're looking at clinical trials, patients in late line therapy that are looking at, for instance, a phase 1 trial, those signals can be really meaningful. So I think busy clinicians understanding that the biology lies in the comprehensiveness is important, and you don't have to have a PhD in genetics, but you need to partner with entities that are going to provide you with actionable insights that tell you as much about that tumor's biology as you can.
Yeah, that makes a ton of sense. And so, one quick myth buster, since we've raised it a little bit, more data does not, of course, automatically improve care. It's only half true. But I love what James said, and we said it in a few different ways. But just think about this: as if you're the physician, go to your bosses, go to your executives, and say, you need something that can help you source the cancer as well as the data that will help you find mainline therapies. And as you start to do this, you'll start to come up with better questions, better experience, and most importantly, faster decisions. So if any of those conversations today surfaced questions on your own, send them to us via email, SMS. We'll get them over to James and the team to make sure they can answer it. If you'd rather stay anonymous and want a way of talking to your executives, so we can do that too. Next time, part 2 of the series, we're going to put liquid biopsy on the table, asking whether a blood draw has actually closed the access gap in precision oncology or just added more to the order set. This is the Shahid Shah Show. Until next time, keep innovating, but stay real and practical like James has shown us today. Thanks a lot, James.
Thank you, Shahid. Pleasure to be here.